Cysteine metabolism refers to the biological pathways that consume or create cysteine. Cysteine is a sulfur-containing non-essential amino acid, meaning the body can synthesize it under normal conditions. Cysteine contains amino group, carboxyl group, and side chain thiol group. Cysteine is important as a precursor for key molecule, detoxification, sulfur metabolism, energy metabolism, and post-translation modification to synthesize protein. Sources of cysteine include high-protein animal-based food and plant-based sources like legumes and seeds. In general, this is the whole process of metabolism cysteine. Now let's move on to transsulfuration pathway. Cysteine synthesis endogenously mainly in the liver, into the lesser extent of the gastrointestinal tract. Cysteine is synthesized from two amino acids, methionine, which is nutritionally essential, and serine, which is not. Methionine furnishes sulfur atoms, and serine furnishes the carbon skeleton. Homocysteine metabolized by the gastrointestinal tract from dietary methionine through transmutation condensed with serine that is catalyzed by cystathionine synthase 2. Form cystathionine. Cystathionine lyase place and break down cystathionine into homoserine and cysteine, and further the deaminate by homoserine deaminase to form alpha-ketobutyrate. Sulfur atoms in the polypeptide chain of cysteine could form a bond together with another cysteine, namely as disulfide bridge by oxidation, then forming cysteine. The significance of synthesis cysteine, it is linking methionine to cysteine, regulates sulfur amino acid metabolism. Next, it is converting homocysteine into cysteine, regulating sulfur amino acid metabolism. Important for antioxidant production, cysteine is a precursor for glutathione, antioxidant. Lastly, act as cellular redox balance, maintaining the cellular balance between oxidation and reduction. Basically, this whole process is happened in the liver. First, the metabolism of cysteine acts as a precursor for glutathione synthesis in the intestinal wall. Inside the intestinal, cysteine is a crucial component in providing a smooth glutathione synthesis. Without cysteine, glutathione can't be produced or synthesized. Cysteine serves as a limiting precursor for GSH synthesis, a critical antioxidant in intestinal cells. While glutathione itself protects intestinal epithelium from oxidative damage and supports detoxification processes. Both important in maintaining cellular redox balance and defending against oxidative damage for optimal glutathione production. The process of metabolism begins with GSH is synthesized in two steps from glutamate, cysteine, and glycine by two enzymes, 1. Gamma-glutamyl-cysteine synthetase. 2. GSH synthetase. GSH controls its production by negative feedback on reaction 1-. If gamma-glutamyl-cysteine is not converted to GSH, it follows an alternate pathway to produce cysteine and 5-oxoproline, which is converted to glutamate via 5-oxoprolinase, reaction 3. The production of H2S begins with cysteine entering cells, where it undergoes enzymatic breakdown. Three key enzymes are responsible for this conversion. The first, cystathionine beta synthase, CBS, converts cysteine into cystathionine and eventually into H2S. CBS operates mainly in the brain and liver, showcasing its systemic importance beyond the gut. The second enzyme, cystathionine gamma lyase, CSE, directly breaks down cysteine into H2S. This enzyme is predominantly found in the gut, liver, and kidneys, emphasizing its role in maintaining intestinal and systemic health. Lastly, 3-mercaptopyruvate sulfur transferase, 3-MST, functions primarily in mitochondria. It works in tandem with an enzyme called cysteine aminotransferase, CAT, which first converts cysteine into 3-mercaptopyruvate, a precursor that 3-MST transforms into H2S. Cysteine serves as a precursor for the synthesis of taurine, a sulfur-containing compound essential for various physiological processes such as bile-salt conjugation and antioxidant defense. This pathway utilizes the sulfur atom from cysteine to regulate the body's sulfur and cysteine balance, thereby preventing the toxic accumulation of these substances. This is the process of taurine synthesis from cysteine. Cysteine is also a precursor for glutathione. The balance between taurine and glutathione synthesis depends on the cellular redox status and metabolic demands. Under oxidative stress, more cysteine is directed toward glutathione production, which may limit taurine synthesis. In intestinal epithelial cells, cysteine is metabolized via the transamination pathway to produce pyruvate, a key intermediate in energy production. This process occurs in situations where the body requires an alternative energy source, such as during fasting, starvation, or oxidative stress. Cysteine sulfonic acid is converted into beta-sulfonylpyruvate, which then breaks down into sulfate and pyruvate. The oxidation of sulfate to sulfite is catalyzed by the enzyme sulfite oxidase, which requires a molybdenum cofactor to function. It also produces pyruvate, which is converted into acetyl-CoA, which enters the citric acid cycle to generate energy. Post-translational modifications involving cysteine. Ribosomes are macromolecular machines, found within all cells, that perform biological protein synthesis. A methylated cap is found at the 5-foot end. The ribosome contains small and large subunits attached together on the long mRNA codons. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site, and inside the ribosome, there are tRNA codons. tRNA is another key molecule, while methionine attached to the end of tRNA corresponds to the August codon. It contains an anticodon that is complementary to the mRNA codon to which it binds. The peptidyl, or P, site and the aminoacyl, or A, site. The methionine is transferred to the A site amino acid. The first tRNA occupies the P site, the second tRNA enters the A site and is complementary to the second codon. The first tRNA exists, the ribosome moves along the mRNA and the next tRNA enters. The basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA. The ribosome moves along the mRNA and the new tRNAs enter. These are the 5 basic processes of the post-translational modification of protein.